Good morning, students. Am I audible?
Yes, ma'am.
Okay. Fine. I hope the screen is visible to everyone. Good morning, my dear YouTube students. So, in the last session, we discussed regarding the concept of microbes in human welfare. Today, we are going to discuss the other most concept in CET as well as NEET. So around 5 or 6 marks where questions will come out of this chapter, biotechnology principle as well as biotechnology applications. ಎರಡು lesson ಕೂಡ ಇದು ಒಂದೇ topic ಅಲ್ಲಿ I will be completing. Okay. So I expect the same participation as of every day from you all, the same kind of fast answers, the same kind of asking the doubts and the interest. Okay. Yes, let us see how many students are on YouTube. Yeah, fine. So let us proceed with the first question of today on biotechnology of Sankalp Learning Solutions crash course. The controlled use of biological agents such as live organisms or enzymes from organisms to produce products and processes useful to human is called as biochemistry, molecular biology, biotechnology, microbiology. The first response is given by
Some biotechnology.
Ravani. Okay, Shashank, please make use of chat box instead of a mic. So Shabina, Jayashri, as well as Swati MS. So most of you answered it correctly. The right answer, very easy question and quick responses from you all. Option C is a right answer. Yes. Next question. The techniques, processes that are included under biotechnology are in vitro fertilization, correcting a defective gene, synthesizing a gene, all of these. Shashank, Pooja, then Swati MS, Shravani Kulkarni, Shabina Sheikh, Aishu. So most of you answered it as option C. Some of you answered it as option D. So the technique or the processes that are included under biotechnology are. So let us check the answer. The right answer is here. Option D, all of this. So here, in vitro fertilization, correcting a defective gene, synthesizing a gene. So all these processes are involved. So option D, all of these is a right answer. Next question. EFB stands for European Federation of Biotechnology. B. Eurasian Federation of Biotechnology. C. East Asia Federation of Biotechnology. D. Ethiopian Federation of Biotechnology. Yes, YouTube students, what is your response on this? Shashank, Shravani, then Swati, Shabina, Aishu answered it. Very good. What about others? Sharada, Jayashri also answered to it. Most of you voted for option A anta. Lakshmi Horkeri is active on YouTube. She answered as option B. So let us check who is right. So the right answer for EFB full form is option A, that is European Federation of Biotechnology. Option A is the right answer. Next question goes like this. Just a minute. Please try to mute yourself once you enter the session. Next question. The definition of biotechnology given by EFB. EFB ಇಂದ ಒಂದು definition ಅನ್ನ ಕೊಡ್ತಾರೆ. Mallikarjun is active on YouTube. Encompasses option A, traditional biotechnology, B, modern molecular biotechnology, C, DNA fingerprinting, D, both A and B. Shashank answered it quickly. Good going. What about others? Shravani Kulkarni, Shabina Shekha is active. Very good. Sharada, Asma. So most of you are voting for option D. Aishu also answered it. Sharath answered it as option C. Great. Yes, what about others? Fine. Yes, YouTube students, what is your answer on this? Swati MS answered it as option D. So the definition of biotechnology given by EFB, European Federation of Biotechnology, encompasses anta ide. Most of you voted for option C. Let us check the answer. The right answer is option D, both A and B. Aden ಮಾಡುತ್ತೆ. Yes, Karthik is active on YouTube. Mallikarjun, Karthik, and Lakshmi Horkeri also answered as option D. Good. So option D, both A and B, that is traditional biotechnology, molecular biotechnology, ಎರಡು ಅನ್ನ encompass ಮಾಡುತ್ತೆ. ಯಾವುದು? EFB definition. So option D is a right answer here. Option D is a right answer. Right. Next question. The two main techniques that gave birth to modern biotechnology are: first one, bioprocess engineering; second one, genetic engineering; third one, human genome engineering; fourth one, molecular biology. Choose the correct option given below. The quick response is given by Shashank, Aishu, Shravani Kulkarni, Tasmiya, and Mallikarjun from YouTube also answered to it. Yes, Rishi, Sharada also answer to it. So most of you voted for option A, bioprocess engineering, genetic engineering. Lakshmi Horkeri answered it as option A. So let us check the answer here. The right answer is, as you all voted, that is option A, bioprocess engineering ಮತ್ತೆ genetic engineering. These are the two main techniques which gave birth to modern technology. Sixth question goes like this. Genetic engineering techniques include: A, altering genetic material; B, sequencing genetic material; C, studying genetic material; D, none of the above. Karthik is active on YouTube. Good. Yes, I got the first response from Swati, Rishi, Shashank, Tasmiya, Aishu, Lubna, Asma, Shabina Sheikh. Good. Sharath, Shravani Kulkarni, Sharada. So most of you answered it as option A anta. Mallikarjun is active on YouTube. So genetic engineering technique include anta ಕೇಳಿದ್ದಾರೆ. Technical ಏನೇನಿರುತ್ತೆ ಅಂತ ಕೇಳಿದ್ದಾರೆ. So the right answer for this is option A, altering genetic material. Genetic material ನ alter ಮಾಡೋದು, change ಮಾಡೋದು. That is the main motto of genetic engineering. Let us proceed to the next question. Gowda is active on YouTube. The specific sequence of DNA that initiate replication. Correct ಆಗಿ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮಾಡ್ಕೊಳ್ಳಿ ಕ್ವಶನ್ ಅನ್ನ. The specific sequence of DNA that initiate replication of alien DNA in our DNA technology is called as initiation sequence. ಆ sequence ಗೆ ಏನಂತ ಕರಿತೇವೆ? Origin of replication, origin of DNA, initiation of DNA. So until I complete reading all the option, please don't answer. Shashank, Sharath answered it. Okay. What about YouTube students? Lubna, Swati, MS answered it. Good. Shabina Sheikh answered it. Tasmia Aishu answered it. Gauda answered it as option C on YouTube. Asma answered it. So the confusion is between option A, option C, option D. So the right answer is here. The specific sequence of a DNA that initiate replication is called as origin of replication. So only few of you voted for option B. Lakshmi Horkeri on YouTube answered it correctly. Option B is the right answer. Eighth question goes like this. Autonomously replicating circular extrachromosomal DNA. Autonomously ಅಂದ್ರೆ self ಅಂತ. A. Vector. B. Capsid. C. Plasmid. D. Bacteriophage. So quick response is given by Shravani, Shabina, Kavya, Swati, Lubna, Shashank, Tasmiya, Sharada, Asma, Sharath, Aishu, Gauda and Karthik on YouTube. Rishi. Okay. ಸಿ ನೋಡಿ ನಾನು ಯಾರ್ದು ಆನ್ಸರ್ಸ್ ಅನ್ನ ಕನ್ಸಿಡರ್ ಮಾಡ್ತೀನಿ ಅಂದ್ರೆ ಒನ್ಸ್ ಐ ಕಂಪ್ಲೀಟ್ ರೀಡಿಂಗ್ ದ ಆಪ್ಷನ್ಸ್ ಯಾವ ಆನ್ಸರ್ ಫಸ್ಟ್ ಬರುತ್ತೆ ಅದನ್ನ ಮೊದಲು ಕ್ವಶನ್ ಕಿಂತ ಮೊದಲೇ ನೀವೇ ಓದಿ ಆಪ್ಷನ್ಸ್ ಅನ್ನ ನೀವು ಹಾಕಿದ್ರೆ ಮೆಸೇಜ್ ಹಾಕಿದ್ರೆ ಐ ವೋಂಟ್ ಬಿ ರೀಡಿಂಗ್ ದಟ್ ಓಕೆ ಸೊ ಪ್ಲೀಸ್ ಮೇಕ್ ಇಟ್ ಕ್ಲಿಯರ್ ಅಂಟಿಲ್ ಐ ಕಂಪ್ಲೀಟ್ ರೀಡಿಂಗ್ ಆಲ್ ದ ಆಪ್ಷನ್ಸ್ ಪ್ಲೀಸ್ ಡೋಂಟ್ ರೆಸ್ಪಾಂಡ್ ಇನ್ ದ ಚಾಟ್ ಬಾಕ್ಸ್ ಸೊ ಮೋಸ್ಟ್ ಆಫ್ ಯು ವೋಟೆಡ್ ಫಾರ್ ಆಪ್ಷನ್ ಸಿ ದಟ್ ಇಸ್ ಪ್ಲಾಸ್ಮಿಡ್ ಅಂತ ಸೊ ದ ರೈಟ್ ಆನ್ಸರ್ ಈಸ್ ಆಪ್ಷನ್ ಸಿ ಪ್ಲಾಸ್ಮಿಡ್ ಸ್ವಲ್ಪ ಜನ ಕನ್ಫ್ಯೂಸ್ ಮಾಡ್ಕೊಂಡಿದ್ದೀರಾ ಡಿ ಅಂತ ಹೇಳಿದ್ರಿ ಸ್ವಲ್ಪ ಜನ ಎ ಅಂತಾನು ವೋಟ್ ಮಾಡಿದ್ದೀರಾ ವಿಚ್ ಇಸ್ ಆಕ್ಚುಲಿ ರಾಂಗ್ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ದ ಕನ್ಸ್ಟ್ರಕ್ಷನ್ ಆಫ್ ಫಸ್ಟ್ ರೀಕಾಂಬಿನೆಂಟ್ DNA was done by using native plasmid of ಫಸ್ಟ್ ರೀಕಾಂಬಿನೆಂಟ್ DNA ಯಾರು ಯೂಸ್ ಮಾಡಿದ್ರು ಅಂದ್ರೆ ಇಟ್ ವಾಸ್ ಯೂಸ್ಡ್ ಬೈ ವಾಟ್ ಈ ಕೊಲೈ ಸ್ಯಾಲ್ಮೊನೆಲ್ಲಾ ಟಿಫಿಮ್ಯೂರಿಯಂ ಬ್ಯಾಸಿಲಸ್ ಥುರಿಂಜಿಯನ್ಸಿಸ್ ಡಿ ಈಸ್ಟ್ meanwhile I will be sharing the notes, CET notes in the YT message box. Do refer it as well as in the chat box of Zoom also I am messaging it. Tomorrow's topic is organism and population. So our CET notes ana ಓದ್ಕೊಂಡು attend agi session ana. So most of you voted it. Let us check the answer. Answer B. Salmonella typhimurium is a right answer. Next question. The first recombinant DNA was rDNA was constructed by Stanley Cohen, Herbert Boyer, Termin and Baltimore. D. Both A and B. Rishi, Shravani, Sharada, Jayashri, Shashank, Shabina Sheikh, Asma, Aishu, Tasmiya. Yes, Kavya. So most of them are voted for option D. Swati, MS, Jyoti. Okay. Few of them, Shailu, voted for option D. Few of them voted for option B. Herbert Boyer anta ಹೇಳ್ತಿದ್ದಾರೆ. So the right answer is here option D. That is Stanley Cohen ಅವರು ಮತ್ತೆ Herbert Boyer ಇಬ್ರು ಕೂಡ the first recombinant DNA ಟೆಕ್ನಾಲಜಿಯನ್ನ DNA ಅನ್ನ ಕನ್ಸ್ಟ್ರಕ್ಟ್ ಮಾಡ್ತಾರೆ ಗೌಡ ಲಕ್ಷ್ಮಿ ಹೊರಕೇರಿ ಕಾರ್ತಿಕ್ ಆನ್ ಯೂಟ್ಯೂಬ್ ಆನ್ಸರ್ಡ್ ಇಟ್ ಕರೆಕ್ಟ್ಲಿ ಸೊ ದ ರೈಟ್ ಆನ್ಸರ್ ಈಸ್ ಆಪ್ಷನ್ ಡಿ ಬೋತ್ ಎ ಅಂಡ್ ಬಿ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ದ ಲಿಂಕಿಂಗ್ ಆಫ್ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ವಿತ್ ದ ಪ್ಲಾಸ್ಮಿಡ್ ವೆಕ್ಟರ್ ಬಿಕೇಮ್ ಪಾಸಿಬಲ್ ವಿತ್ ಯಾವ ರೀತಿ ಲಿಂಕ್ ಮಾಡಿದ್ರು ಯೂಸಿಂಗ್ ವಾಟ್ ಅಂತ ಎ DNA ligase, B RNA ligase, C DNA polymerase, D RNA polymerase. Jayashri, Shashank, Aishu, Kavya, Rishi, Shailu. Okay, the fight is between A and D. Asma, Shabina, Sheikh. Yes, YouTube students, what is your response on this? Jyoti, Swati, MS. Fine. So most of you voted for option A, few of you voted for option D, that is RNA polymerase. As they asked the linking of antibiotic resistance gene. So the right answer will be DNA ligase. DNA ligase. Karthik answered it as option C. So the right answer is option A. 12th question goes like this. The different basic steps of genetically modifying an organism are given below randomly. Okay, just a minute. Actually, this question is not completed because the options are not there. Let us skip this. Uh, okay, next question. The key tools, just a minute. Who got? Hmm. The key tools required for the recombinant DNA technology are restriction enzymes, polymerase enzymes, ligases, vector, host organism. So, ಯಾವ್ಯಾವ ಬೇಕಿದ್ರಲ್ಲಿ? So, the options are not given. ಆಪ್ಷನ್ ಡಿ ಅಂದ್ರೆ ಆಲ್ ಆಫ್ ದೀಸ್ ಎಲ್ಲವೂ ಬೇಕು ನಮಗೆ ಹೌದಾ ಸೊ ಎಲ್ಲವೂ ಬೇಕಾಗತ್ತೆ ಫೈನ್ ಸೊ ಆಪ್ಷನ್ ಡಿ ಅಂತ ಇದೆ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್ ದ ಎನ್ಸೈಮ್ಸ್ ಕಾಮನ್ಲಿ ಯೂಸ್ಡ್ ಇನ್ genetic engineering are restriction endonuclease and polymerase, endonuclease and ligase, restriction endonuclease and ligase, ligase and polymerase. So, which are the enzymes commonly used in genetic engineering? Rishi Jyoti answered it. Sharat Aishu answered it. Kavya answered it. Tasmiya Banu. Shashank yes answered to it. What about YouTube students? Be fast. Jyoti Laddi answered it. So the vote is for option C, that is restriction endonuclease and ligase, exactly the right answer. The first restriction endonuclease to be discovered is first restriction endonuclease andre enu? Yes, YouTube students Karthik, Lakshmi Horkeri, Mallikarjun are active and answered it as option A. So 15th question, the first secretion endonuclease to be discovered is so Rishi, Aishu, Sharat, Swati, Asma, Shashank, Bhuvan, Shabina Sheikh, Sharada. So most of you are answering for option A. Few of you are answering for option B. So the right answer is here option B, that is hind 2. Hind 2 is a first restriction endonuclease enzyme. Lakshmi Horikeri answered it as option C. No, 15th answer is option B, that is hind 2. Just a minute. Next question. In recombinant DNA technique, the term vector refers to a donor DNA. It is identified and picked up through electrophoresis. Plasmid transfers DNA into host cell. Collection of entire genome in the form of a plasmid. Enzyme cuts the DNA at specific sites. The quick response is given by Shashank, Kavya, Tasmiya Banu, Swati, Jyoti, Sharat. Mallikarjun is active on YouTube. Yes, YouTube students, you can please like and subscribe to our channel and the video. Asma, Rishi also answered it. So the right answer for this is very good. Option B, Lakshmi Horkeri also answered to it. Plasmid transfers DNA into host cell is a right answer. Which of the following is used in recombinant DNA technique? Cell wall of virus, gene which produces capsid of virus, bacteriophage, capsid of virus. Which of the following is used in rDNA technology? Shashank, Shabina, then Aishu, Tasmiya Banu, Kavya H, Rishi, Jyoti, Sharat. Most of them voted. Yes, what about others? Be fast. Asma also answered it. So the right answer for this is option number C, that is bacteriophage. During gene cloning, which is called as gene taxi, A, vaccine, B, plasmid, C, bacteria, D, protozoa. Lakshmi Horkeri answered it as option B. Rashmi from YouTube answered it as option B. Swati, Aishu, Sharat, Kavya, Tasmiya, Banu, Shashank. Okay. So most of you answered it as option B. Some of you voted for option D. Lakshmi Horkeri answered it as option B. Jyoti, option B. Okay. So the right answer for this is option B, that is plasmid. What about others? YouTube students, be fast, be active. Next question. Which of the following is not a feature of the plasmid? ಯಾವುದು ಪ್ಲಾಸ್ಮಿಡ್ನ ಗುಣಲಕ್ಷಣ ಅಲ್ಲ? A. Circular structure. B. Transferable. C. Single stranded. D. Independent replication. Tasmiya, Banu, Aishu, Shashank, Shailu. Yes, Kavya H. Jyoti. Sabina Sheikh. Sharat. So most of them are voted for option C, that is single stranded ಅನ್ನೋದು. It is not a feature of plasmid ಅಂತ. So the right answer is here, that is option C. Circular ಇರುತ್ತೆ ನಮ್ಮ ಪ್ಲಾಸ್ಮಿಡ್, ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡುವ ಹಾಗಿರುತ್ತೆ, ಇಂಡಿಪೆಂಡೆಂಟ್ ಆಗಿ independent ಆಗಿ replicate ಆಗತ್ತೆ. So option C is a right answer. Next question. Which vector can only clone only a small fragment of DNA? ಯಾವ ವೆಕ್ಟರು ಸ್ಮಾಲ್ ಫ್ರಾಗ್ಮೆಂಟ್ ಆಫ್ DNA ಅನ್ನ ಕ್ಲೋನ್ ಮಾಡತ್ತೆ? ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಆರ್ಟಿಫಿಷಿಯಲ್ ಕ್ರೋಮೋಸೋಮ್, ಈಸ್ಟ್ ಆರ್ಟಿಫಿಷಿಯಲ್ ಕ್ರೋಮೋಸೋಮ್, ಸಿ ಪ್ಲಾಸ್ಮಿಡ್, ಡಿ ಕಾಸ್ಮಿಡ್. Yes, Lakshmi Horkeri is active on YouTube. Good. Aishu, Kavya H, Shabina Sheikh, Shashank, Tasmiya, Sharat. Yeah. For the Zoom students, whenever you are chatting, so chat box ಅಲ್ಲಿ ಎಂಟರ್ ಆದ್ಮೇಲೆ ಮೆಸೇಜ್ ಟು ಅಂತ ಇರುತ್ತೆ. ಮೆಸೇಜ್ ಟು ನಿಧಿ ಮ್ಯಾಮ್ ಅಂತ ಮಾಡಿ, ಸೊ ದಟ್ ಯುವರ್ ಆನ್ಸರ್ ವಿಲ್ ಬಿ ವಿಸಿಬಲ್ ಓನ್ಲಿ ಟು ಮೀ. ಎಲ್ರಿಗೂ ನಿಮ್ಮ ಆನ್ಸರ್ ಹೋಗೋದು ಬೇಡ. ಸೊ ಯು ಕ್ಯಾನ್ ಡೂ ಇಟ್. ಓಕೆ, ಜಸ್ಟ್ ಅ ಮಿನಿಟ್. So here, which vector can alone only a small fragment of DNA? Bacterial artificial crop. 1 minute. Please take care to mute yourself. ಒಂದೊಂದ್ಸಲ ಮೈಕ್ ನಿಮಗೆ ಗೊತ್ತಾಗದೆ ಹೋಗಿ ಆನ್ ಆಗಿರುತ್ತೆ. So we can hear all the things which is going on at your home. Okay, so which vector can clone only a small fragment of DNA? A is bacterial artificial chromosome, B is yeast artificial chromosome, C is plasmid, D is cosmid. Sharat, Shashank, Akash is active on YouTube, Lakshmi is active, Shailu, Tasmiya, Banu, Aishu also answered to it. Let us check the answer. The right answer is here, option C, that is plasmid. Next question. Just a minute. Okay. Identify A, B, C, and D in the given diagram of E. coli cloning vector PBR322. Okay, so here A, B, C, D are given. You have to identify. Read out the options and let me know the answer. Yes, YouTube students, be fast. Very quick responses from Zoom students. Shabina Sheikh, Aishu, Shashank, Swati, Shailu, Jyoti. So most of you voted for option A anta. Kavya and Sharada, Lakshmi Horkeri on YouTube voted for option A. Tasmiya, Jyoti also answered to it. So the right answer is here option B, E. coli, B, Bam, HI, HR, C, Ori, D, Ampar. Right? Next question. Hmm. Hmm. This is kind of a different question. The given figure is a diagrammatic representation of the vector PBR322. Which one of the given option correctly identified its certain components? ORI andre original restriction enzyme, ROP andre reduced osmotic pressure, hind 3 E. coli are selectable markers, AMPAR TET are andre antibiotic resistant gene. So which is a correct component of it? Yes, Akash, Mallikarjun, Lakshmi are active on YouTube and they voted as option A. Aishu, Shailu, Shashank, Jyoti, Shabina Sheikh, Rashmi on YouTube, Lakshmi Horkeri, Swati MS, Mallikarjun, Kavya H. So most of you voted for option A, few of you voted for option C. So the right answer is option D. AMPAR, TET-R are the antibiotic resistant genes. ಅನ್ನೋದು ಸರಿಯಾದ ಉತ್ತರ. Next question. The two antibiotic resistance genes on vector PBR322 are for ಏನನ್ನ indicate ಮಾಡ್ತವೆ ಅವು? Ampicillin and tetracycline, ampicillin and chloramphenicol, chloramphenicol and tetracycline, tetracycline and kanamycin. Yes, Sharada, Aishu, Shailu, Akash from YouTube answer to it. Shashank, yes. Be fast, be fast. Tasmiya Banu, Swati MS. So most of you answered it as option C ಅಂತ ಹೇಳ್ತಿದ್ದೀರಾ. Kavya and Jyoti. Okay. So the right answer to it, Lakshmi Varsha Kolkar is active on YouTube. So option A is the right answer. C ಅಲ್ಲ. Ampicillin ಮತ್ತೆ tetracycline. C ಏನಿದೆ chloroamphenicol and tetracycline. C is a wrong option. So Lakshmi Horkeri also answered to it. Mallikarjun answered to it. Okay. The function of ORI in a vector. ORI ಕೆಲಸ ಏನು vector ಅಲ್ಲಿ? Help in replication of linked DNA, control copy number of linked DNA, help in selecting recombinants. D, both A and B. Both A and B. Yes, YouTube students. Swati, Shashank, Kavya, Aishu, Jyoti, Tasmiya Banu. Yeah, most of you answered it as option A. Shabina Sheikh, option A. So few of you voted for option D. Both A and B anta. So the right answer is option D. That is help in replication of linked DNA, control copy number of the linked DNA. Lakshmi Horkere exactly right. Option D is a right answer. Bhavya also answered it as option A. No, it is option D. Both A and B. Next question. A gene whose expression helps to identify transformed cells is known as, what is that called as? Selectable marker, B. Vector. C. Plasmid. D. Structural gene. Yes, Varsha Kolkar, Mallikarjun, Lakshmi is active on YouTube. So I got quick responses from Aishu, Shashank, Swati MS, Jyoti, Bhavya, Tasmiya, Shailu. So all of them voted for option A. Kavya H also voted for option A, that is selectable markers ಅಂತ ಹೇಳ್ತಿದ್ದೀರಾ. ಅವ್ರು ಕೇಳಿದ್ದು expression to identify transformed cells ಯಾವುದು ಅಂತ. Okay, fine. So the right answer is here. As you all selected, option A, selectable marker is a right answer. Next question. A selectable marker is used to. Yes, Lakshmi and Varsha, you are right. Option A is the right answer for the previous question. Rashmi is also active on YouTube. A selectable marker is used to. ಯಾಕೆ ನಾವು selectable marker ಅನ್ನ ಯೂಸ್ ಮಾಡ್ತೇವೆ? Read out the options and tell me the answers. Yes. Do like and subscribe to our channel, you YT students. Do like the video, comment the video so that we will get to know your suggestions. Shashank also answered to it. Good. Aishu, Sharat, Tasmiya, Shabina, Jyoti. Fine, let us check the answer. The right answer is here. Option A, that is, it helps in eliminating the non-transformants so that transformants can be regenerated. Option A is a right answer. Next question. In recombinant DNA carrying antibiotic resistance gene, example ampicillin anta ankolona, is transferred into E. coli cell. The host cell is transformed into ampicillin resistant cell. The ampicillin resistant gene in this case is called as A. Vector, B. Plasmid, C. Selectable marker, D. Cloning sites. Read out the question properly. Sharat answered it quickly. Mallikarjun and Lakshmi are active on YouTube. Jyoti, Shashank, Jyoti Natekar, Tasmiya Banu, Kavya H. So the fight is between option C and option D. Shravani Kulkarni. So the right answer is here. Option C, selectable marker. Ampicillin, tetracycline, selectable marker ಆಗಿ ನಾವು ಯೂಸ್ ಮಾಡ್ತೇವೆ. Next question. The different steps involved in the process of recombinant DNA technology are given below randomly. Random ಆಗಿ ಕೊಟ್ಟಿದ್ದೀವಿ ಹಿಂದೆ ಮುಂದೆ. So arrange these in correct order ಅಂತ ಹೇಳಿದ್ದಾರೆ. So the steps are extraction of a desired gene product. Second, amplification of the gene of interest. Third, isolation of a desired DNA fragment. Fourth, ligation of the DNA fragment into a vector. Fifth, insertion of recombinant DNA into the host. So you have to select the correct order. A, B, C, D are given. Select the correct order and let me know the answer. Varsha Kolkar, Kadrolli Mahesh, Mallikarjun are active on YouTube. Good. Shashank HS answered quickly. Aishu answered to it. Shravani, Tasmiya, Bhavya, Swati MS. So whether anyone among yourself, that is Zoom students and YouTube students, anyone are attending NEET Google Forms? ಯಾರಾದ್ರೂ NEET Google Forms attempt ಮಾಡಿದ್ರಾ? Kavya, Jyoti also answer to it. Anyone? ಯಾರಾದ್ರೂ attempt ಮಾಡಿದ್ರಾ NEET Google Forms ನ? Okay. Shashank answered it as ನಾವು ಇವತ್ತಿಂದ ಮಾಡ್ತೀವಿ ಅಂತ. I will do from today. Okay. So do you have that link? I pasted in the chat box. Okay, fine. ಅದನ್ನ ಓಪನ್ ಮಾಡಿ ಸೇವ್ ಮಾಡಿ ಇಟ್ಕೊಳ್ಳಿ. Then that time it will be open. And if you want that same link will be shared in the WhatsApp group. Otherwise, if you go to our channel, ಸೊ ಈ ವಿಡಿಯೋ ಇದೆ ಅಲ್ವಾ, ಇವತ್ತಿನ ವಿಡಿಯೋ ಬಯೋಟೆಕ್ನಾಲಜಿ, ಅದರ ಡಿಸ್ಕ್ರಿಪ್ಷನ್ ಬಾಕ್ಸ್ ಅಲ್ಲೂ ಕೂಡ ಯು ವಿಲ್ ಬಿ ಹ್ಯಾವಿಂಗ್ ದಟ್ ಲಿಂಕ್. ಸೊ ದ ರೈಟ್ ಆನ್ಸರ್ ಈಸ್ ಆಪ್ಷನ್ ಬಿ, ದಟ್ ಈಸ್ ಸೆಕೆಂಡ್, ಥರ್ಡ್, ಫೋರ್ತ್. See, out of 20 students, only one student responded regarding the Google Forms. What about YouTube students? Anyone attended Google Form? So, third one, isolation of a DNA, desired DNA fragment. Second, amplification of the gene product. Fourth, that is ligation of DNA fragment into a vector. Fifth, insertion of recombinant DNA into the host. First, extraction of the desired gene of interest. Exactly, option B is the right answer. Some of them got confused with option C. In bacterial cells, the membrane is digested with the help of an enzyme, cellulase, lysozyme, chitinase, lipase. Shashank, Tasmia. ಸಿ ನೀಟ್ ಗೂಗಲ್ ಫಾರ್ಮ್ಸ್ ಅಲ್ಲಿ ನೀಟ್ ಕ್ವಶನ್ಸ್ ಇರುತ್ತೆ. ನಾನ್ ಯಾಕೆ ನೀಟ್ ಬರೀಲಿ, ನಾನು ಅಟೆಂಡೇ ಆಗ್ತಿಲ್ಲ ನೀಟ್ ಎಕ್ಸಾಮ್ ಅಂತ ತಿಳ್ಕೊಬೇಡಿ. ದೇರ್ ಮೈಟ್ ಬಿ ಅ ಚಾನ್ಸ್ ದಟ್ ದ ನೀಟ್ ಕ್ವಶನ್ ದೇ ಕ್ಯಾನ್ ಬಿ ಆಸ್ಕ್ಡ್ ಇನ್ ಅವರ್ ಕರ್ನಾಟಕ ಸಿಇಟಿ ಆಲ್ಸೋ. So please do attend NEET questions or NEET Google Forms. Don't worry, your marks will not be revealed. Only you will be knowing your marks and how your performance is. Okay, please do attend the NEET Google Forms. Swati MS, Bhavya, Varsha Kolkar, Mallikarjun are active on YouTube. So the right answer here is option B, that is lysozyme ಅನ್ನೋ enzyme ಏನ್ ಮಾಡುತ್ತೆ, it helps in the digestion of a membrane. Next question. RNA is removed by the treatment with option A, ribonuclease, B, protease, C, chitinase, D, cellulase. RNA is removed by the treatment with. Aishu answer to it. Shashank, yes. Shravani Kulkarni, Tasmiya, Sharat. Bhuvan. So most of them answered it as option A, that is ribonuclease is a right answer. Very easy question. Proteins are removed by the treatment with ribonuclease, chitinase, cellulase, protease. Varsha Kolkar and Mallikarjun are active on YouTube. Yes, be fast, YouTube students. Shashank, Tasmiya Banu, Bhuvan, Aishu. So some of them asked NEET Google form in the WhatsApp. See, you will be having the same thing in the YouTube link also. Okay, so you can go through it. WhatsApp ಅಲ್ಲಿ ಒಂದೊಂದ್ಸಲ ಬರುತ್ತೆ, ಒಂದೊಂದ್ಸಲ ಬರಲ್ಲ. ಡ್ಯೂ ಟು ಅ ಲಾಟ್ಸ್ ಆಫ್ ಮೆಸೇಜಸ್, ಯಾವುದೋ ಒಂದು ಮೆಸೇಜ್ ಅಲ್ಲಿ ಮುಚ್ಚೋಗಿರುತ್ತೆ. ಸೊ ಝೂಮ್ ಕ್ಲಾಸ್ ನಡೆಯುವಾಗ ಐ ವಿಲ್ ಬಿ ಪೇಸ್ಟಿಂಗ್ ದ ಲಿಂಕ್ ಇನ್ ದ ಚಾಟ್ ಬಾಕ್ಸ್. ಸೊ ಅವಾಗಲಾದ್ರೂ ನೋಡ್ಬೋದು ಅಥವಾ ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಾದ್ರೂ ನೋಡ್ಬೋದು. So the right answer for this is very easy question, option D, protease. DNA precipitation out of a mixture of biomolecules can be achieved by the treatment with. Applied question. A, chilled ethanol, B, methanol at room temperature, C, chilled chloroform, C, isopropanol. DNA precipitation yavaga agatte, it is achieved by a treatment with. The quick response is given by Sharat, Aishu, Shashank, Kavya. Swati MS. So most of them are voted for option A, few of them voted for option D. So the right answer is here option A, that is chilled ethanol ಅನ್ನೋದು ಸರಿಯಾದ ಉತ್ತರ. Next question. Purified DNA ultimately precipitate out, and this can be seen as a collection of fine threads in the suspension as seen in the figure. It refers to ಆ ಒಂದು ಸಸ್ಪೆನ್ಷನ್ ಗೆ ನಾವೇನಂತ ಕರಿತೇವೆ? DNA spooling, DNA digestion, DNA recognition, DNA bands. Shashank, Sharada, Mallikarjun is active on YouTube. Good. Kavya H, Aishu, Swati MS. Okay. So most of them voted for option A, that is DNA spooling ಅನ್ನೋದು ಸರಿಯಾದ ಉತ್ತರ. Chimeric DNA is chimeric DNA ಗೆ ಏನಂತ ಕರೀತಾರೆ? A gene clone. What it is? Recombinant DNA, transpool, vector shuttle. What it is called as? Chimeric DNA is what it is? ಬರ್ತಾ ಬರ್ತಾ ಅಪ್ಲೈಡ್ ಕ್ವಶನ್ ಜಾಸ್ತಿ ಆಗತ್ತೆ. ಐಶು, ಕಾವ್ಯ, ಸ್ವಾತಿ, ಶಶಾಂಕ್, ತಸ್ಮಿಯಾ, Mallikarjun on YouTube. Most of you voted for option B, recombinant DNA anta. So the right answer is recombinant DNA. Next question. Polymerase chain reaction needs. ಏನ್ ಬೇಕಾಗತ್ತೆ polymerase chain reaction ಗೆ? DNA template, primers, stack polymerase, all of these. Polymerase chain reaction needs. Varsha Kolkar is active on YouTube. Swati, Kavya, Aishu, Shashank, Bhuvan, Tasmiya Banu, Swati. So most of you voted for option D, all of these anta. The right answer is all of these. Ellavu beku. Primers are, what are primers? The options are given below. Small chemically synthesized oligonucleotides of about 10 to 18 nucleotides. B. Chemically synthesized oligonucleotides about 10 to 18 nucleotides that are not complementary to the region of template DNA. C. The double-stranded DNA that need to be amplified. D. Specific sequences present on recombinant DNA. What are primers? Swati MS answered to it. Mallikarjun, Kadroli Mahesh, Varsha are active on YouTube. Kavya H, Shashank answered to it. Aishu answered to it. So the right answer for this question is option A. Small chemically synthesized oligonucleotides of about 10 to 18 nucleotides that are complementary to the region of template DNA. So most of them answered to it. Some of them got confused with option C. The Taq polymerase enzyme is obtained from Theobacillus ferrooxidans, Bacillus subtilis, Pseudomonas subtilis, Thermus aquaticus. Yes, Nazma is active on YouTube. So the right answer for the question is Kavya, Shashank, Aishu are active. Swati. So some of you are voting for option B, some of you are voting for option D. So the right answer is here option D, that is Thermus aquaticus. Next question. A single PCR amplification cycle involves A. Denaturation, B. Extension, C. Annealing, D. All of these. Shravani, Tasmiya, Kavya, Shashank. Most of them voted for option D. Mallikarjun, Varsha Kolkar are also voting for the question. From the YouTube, Swati M, Shravani Kulkarni. Okay, so the right answer is here option D, that is all of these. Denaturation iratte, extension iratte, annealing iratte. The correct order of steps in PCR: denaturation, extension, annealing, annealing, extension, denaturation, extension, denaturation, annealing, denaturation, annealing, extension. Which is the correct order? Yes, YouTube students, be fast. Shravani, Swati, Shashank, Aishu, Shabina, Jyoti, Tasmiya Banu. Yes, YouTube students, be fast. Swati MS. So most of them voted for option D, denaturation, annealing, extension anta. So during our theory classes, I have given you a hint. DAE anta ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. Day anta ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. D for denaturation, A for annealing, E for extension. So option D is a right answer. Next question. See to the diagram properly. Okay. The below diagram refer to PCR. Identify the steps A, B, C, and D and select the correct option. Option A is here. Denaturation at 94 to 96 degrees Celsius is A. B. Annealing at 40 to 60 degrees Celsius. C. Extension though tack polymerase. D is amplified. ಇಲ್ಲಿದೆ. A, B, C, D ಇಲ್ಲಿದೆ. So, ಆಮೇಲೆ B. These are the options. A option is here. These are B, C, D options. Look to the options and let me know the answer. This is A, this is B, this is C, this is D. Shashank, yes, answered to it. Mallikarjun and Varsha Kolkar as active on YouTube. Tasmia Banu, Shabina Sheikh, Swati MS, Bhuvan. Yes. Bhavya. So the right answer for this question is option A, denaturation, annealing, extension, amplified. Okay, next question. If recombinant DNA bearing gene for ampicillin resistance is transferred into E. coli and the host cells are spread on agar plates containing ampicillin, then what happens? Both transformed and untransformed recipient cells will die. Both transformed and untransformed recipient cells will grow. Transformed recipient cells will grow. Untransformed recipient cells will die. Transformed recipient cells will die. Untransformed cells will be growing. What happens when you, what we can say, ampicillin resistant gene will be transferred? Kavya H answered to it. Nazma, Mallikarjun are active on YouTube. Shashank, yes, answered to it. Tasmia Banu answered to it. So the right answer is option C. Transformed cells will grow, untransformed cells will die. ಒಬ್ಬೊಬ್ರು ಡಿ ಅಂತ ಹೇಳಿದ್ರಿ. ಡೋಂಟ್ ಗೆಟ್ ಕನ್ಫ್ಯೂಸ್. ಯಾವ ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ ಆಗಿರ್ತವೆ ಅವು ಮಾತ್ರ ಉಳ್ಕೋತವೆ. ಟ್ರಾನ್ಸ್ಫಾರ್ಮ್ ಆಗದೆ ಇರೋದು ಸತ್ತು ಹೋಗ್ತವೆ. ಸೊ ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. ಪ್ರೋಟೀನ್ ಎನ್ಕೋಡಿಂಗ್ ಜೀನ್ ವಿಚ್ ಈಸ್ ಎಕ್ಸ್ಪ್ರೆಸ್ಡ್ ಇನ್ ಹೆಟರೋಲೋಗಸ್ ಹೋಸ್ಟ್ ಈಸ್ ಫಾರಿನ್ ಪ್ರೋಟೀನ್, ಹೆಟರೋಲೋಗಸ್ ಪ್ರೋಟೀನ್, recombinant protein, alien protein. Bhavya answer to it. Varsha Kolkar, Mallikarjun are active on YouTube. Aishu, Kavya, Bhavya, then Shashank, Tasmia Banu. So most of the votes are for option C, recombinant protein anta. Shabina Sheikh. So the right answer is here option C. It is called as recombinant protein. In a continuous culture system ಅಲ್ಲಿ ಬಂದಾಗ continuous culture system ಅಲ್ಲಿ used medium is drained out. ಯೂಸ್ ಮಾಡಿರುವಂತಹ medium ಅನ್ನ drain ಮಾಡ್ತೇವೆ. Biomass produced is high. No new medium is added. Both A and B. Yes, Varsha Kolkar answer to it. Shashank, yes, answer to it. Jyoti, Shabina Sheikh, Kavya, Aishu, Tasmia. So the right answer for this question is option D, both A and B. Next question. A bioreactor is hybridoma, cultures products containing radioactive isotope, cultures for the synthesis of new chemicals, cultures large volume of cells. Yes. Kavya answered it quickly. Swati MS, Shabina Sheikh, Jyoti, Shashank, yes. Tasmiya Banu, Aishu. So the right answer is here. Option D. That is culture's large volume of living cell is a right answer. Next question. Stirred tank bioreactor have been designed. ಬಯೋರಿಯಾಕ್ಟರ್ ಅಲ್ಲಿ ವಿಧವಿದೆ ಹೌದಾ, ಎರಡು ವಿಧಗಳಿವೆ. ಸ್ಟಿರ್ಡ್ ಟ್ಯಾಂಕ್ ಬಯೋರಿಯಾಕ್ಟರ್ ಎದಕ್ಕೆ ಡಿಸೈನ್ ಮಾಡಿದ್ದಾರೆ? ಪ್ಯೂರಿಫಿಕೇಶನ್ ಆಫ್ ದ ಪ್ರಾಡಕ್ಟ್, addition of preservative to the product, availability of oxygen throughout the bioreactor, ensuring anaerobic respiration in the culture vessels. Swati, Shashank answer to it. Bhima Bai Ambedkar is active on YouTube. Bhavya, Tasmiya, Shabina Sheikh answer to it. Jyoti answer to it. So the right answer for this question is option C, that is availability of oxygen. Next, stirred tank bioreactors are advantageous over shake flask. ಯಾಕೆ stirred tank bioreactor? So advantageous ಅಂತ. Next question, just a minute. A. Provide high temperature and pH. B. Provide better aeration and mixing property. Do not allow the entry of carbon dioxide. Easy to operate. Bhavya Hanchinal answer to it. Varsha and Mallikarjun are active on YouTube. Shabina Sheikh answer to it. Tasmiya, Aishu, Swati MS, Shashank. So the right answer for this is option B, provide better aeration. Last question of today about biotechnology. Simple stirred tank bioreactor is given below. Identify A, B, C, D, E. A, B, C, D, E. Options are here. See to the options and let me know the answer. Varsha Kolkar is active on YouTube. Yes, very good responses from everyone. So the right answer for this is option A. Motor. A is motor, B is foam sticker, sorry, foam breaker, C is sterile air, D is acid steam for sterilization, E is acid base for pH control. So option A is the right answer. Now all of you please stay tuned in so that I will share what you can say uh, the Google form as well as CET links. Tomorrow's this one, organism and population. Tomorrow topic is organism and population. So I request everyone to please attend the Google forms. It will help you for CET also. Yes. I'm pasting Google form link. YouTube students, you can also make use of it. Fine? Fine. So let me end up the session here. Have a nice day. Take care. Bye-bye.